So in the past couple of chapters, we've talked about adaptive immune system. We've talked about innate immune system. We talked about issues with the immune system, whether it's active, hyperactive, too sensitive, or whether it's attacking self, or it's just deficient. Today, we're going to talk about how we can basically influence our immune systems to protect us from various pathogens without actually experiencing the disease. Okay, so where vaccinations started was back in the 1500s in China. There was, I believe it was a woman that she wasn't technically a doctor, maybe I should have read up on it, but they started taking powder from the scabs of smallpox victims, and then the person would blow the powder into the patient's nose, and that would kind of protect them from getting more virulent or more deadly forms of smallpox. And this process was called variolation. And you're basically giving them the actual virus. Okay, but it was dried out and given in much smaller concentrations than normal. And so this would result in milder infections, maybe less deadly infections, and decrease the mortality rate down to 1 to 2%, which is still rather high. But this process of using the immune system and kind of training it to help us overcome certain diseases started back in the 1500s. And in England, they didn't like the 1 to 2% death rate. That was too much for them. And remember our golden age of microbiology, where we had all of these new advances and things like that. Well, just about 75 to 100 years before then, in 1796, Mr. Edward Jenner made an observation, and I think we talked about him on the first day of class, where he noticed that milkmaids, women that their employment was to go and milk cows, they would sometimes get a disease known as cowpox from the cows because they had so much contact physically with the cows during the milking and the feeding process and all of that. And Jenner noticed that these milkmaids very rarely got smallpox, or if they did get smallpox, it was pretty mild. And so he came up with the idea that maybe smallpox and cowpox are just close enough to each other that maybe if we could give people cowpox, that will protect them from smallpox. So Edward Jenner purposely not inoculated or gave a boy cowpox. Okay, he deliberately exposed the boy to cowpox. Boy got cowpox, which isn't nearly as serious as smallpox. He recovered, let the boy recover, return to health, all of that kind of stuff for a while, and then he infected the boy with smallpox. And so if Jenner was wrong, the boy could have definitely died, but the boy showed no symptoms of smallpox, and this was basically the first kind of experiment with the process of vaccination. And vaccination comes from the Latin word vacca. The Spanish word has one C, this one has two, and that's because of its link with cows. So that's how vaccination got its name. Then further into the microbiology golden age, Louis Pasteur developed a couple of the first vaccines. He developed a vaccine against anthrax to help local farmers protect their cattle against a deadly disease. And he also became one of the first inventors, or the first to come out with a rabies vaccine to protect humans from rabies. and during that golden age of microbiology, as scientists became more and more aware of which pathogens caused which certain diseases, scientists started developing more and more vaccines. And currently, there are over 25 vaccines or 25 infections that there are vaccines for. And here's some of them. I'm not going to make you memorize all of these. These are just kind of a FYI here. So anthrax, that's not something that normally we would be given to the general population in the US. BCG, the tuberculosis vaccine, again, not something routinely done here in the US unless you're traveling to an endemic area. Same with cholera. The Hib, Haemophilus influenzae B, is one that we routinely give. Hep A and B, HPV, influenza, MMR, measles, mumps, rubella, those are routine. Meningococcal, against meningococcal meningitis. If you live in the dorms, you likely had to get that vaccination before you could move in. Pneumococcal vaccine for elderly patients. Let's see, polio. If you go into veterinary medicine or work as a zookeeper, you'll likely need to get a rabies vaccine, but general population, not. Rotavirus, which is a GI disease, illness, diarrhea, that type of thing. Smallpox vaccine, that's not really given anymore. If probably for most of you guys, your grandparents, but maybe some of your parents that are older than I am, have a smallpox vaccination scar. Back in the 50s, 60s, maybe early 70s, they gave a smallpox vaccine in the arm. And basically it was a circular shaped instrument with prongs on it, and it would leave a scar on the arm. It was deposited into the skin. But that we'll talk about that in a second, but we have eradicated smallpox, and so we no longer vaccinate for that. Let's see, chickenpox vaccine, and the herpes zoster is going to be the shingles vaccine. Those are the ones that we typically would see here in the US. So Edward Jenner protected the boy against smallpox using the cowpox vaccination. But we don't see smallpox anymore because it has been eradicated. And in 1977 in Somalia, that was the last known case of smallpox on Earth. And one thing that made smallpox a good candidate for eradication was the fact that it does not enter into animal populations. It's only a human virus. And so that was helpful because, it's impossible to control animal movements and disease spread and things like that. Also, as you can see, the signs of the disease are very clear, the pustules, so you positively know which people have it and which ones do not. And so there were some other things that went into that to make smallpox a really good candidate for eradication. And it took some very intentional, concerted efforts of many different countries and all sorts of people in different walks of healthcare to get this disease eradicated, but they have. And that's what our case study is going to be about this afternoon. You'll read through that. It talks about where some stockpiles of smallpox are used for research procedures. And so you'll have an opportunity to think through the ethics on some of those things. So currently, the CDC estimates between 1994 and 2013 in the US, vaccines prevented 21 million deaths, hospitalizations, and over 700,000 deaths. I don't think this book, looks at some of the graphical data based on vaccinations. And you know what, let me just pull this up here for just 1 second. Let me, let's see, stop sharing that and share this. Let's see if we can find something here. Let's see. Oops, come back. Okay, so here is reported paralytic polio cases and then deaths, which is, cases are in blue, deaths are in red. Now, let's see. This red line here, this is number of cases. This red line here, 1955, that's when they first introduced the polio vaccine. And look, almost immediately, the number of polio cases in the US dropped to very close to zero. So there is a very big effect of vaccination on the number of cases and number of deaths of many different diseases. Okay, so I just wanted to show you that pictorially. I think it's kind of interesting. All right, so let me pick back up here, share screen. All right. So even though vaccines are life-saving, basically since vaccination began, people have really kind of pushed back against it. There's always been some controversy with that. Even in the 1800s, there was controversy about the use of vaccinations. There were also groups of Hindus that were upset because the cow is sacred to their religion and they were using cattle for the vaccination process, and they didn't want to inject cattle parts into them. And, you know, there's always been some controversy based on, you know, whether or not we should be vaccinating people or not. Okay, we'll talk a little bit more of that here in just a second, but we have this slide here talking about eradication of smallpox. Polio is the next disease on the list to eradicate. We're not there yet, but they're working towards completely eradicating polio across the world. The US is pretty much a polio-free country because of vaccination efforts and hygiene and many different other factors. But the most recent, it says re-eruption of fears about vaccination started out in 1998 in a paper published in The Lancet. And The Lancet is basically a medical journal or a peer-reviewed journal out of London, I believe, or somewhere in England. And they published a paper in this, you know, highly technical, peer-reviewed publication that had 12 patients in their study, which is very low, and claimed that there's a correlation. Remember, correlation doesn't mean causation. A correlation between the MMR vaccine and the development of autism. So you've probably heard somewhere on the internet or from friends or family members that vaccines have been linked with autism. And that statement is true. There was a link, there was a correlation made between MMR and autism, but this study was highly flawed. There were only 12 patients there, and the Lancet in 2010 fully retracted this study because it had bad science. And one of the reasons was, besides there were only 12 subjects, is that the authors, the people conducting the study, were receiving their paycheck from lawyers that were hired by parents whose children had become autistic, or were diagnosed as autistic, I should say, and they were trying to sue vaccine companies. So there was a major conflict of interest. The author, or the primary author of the study, lost his medical license and had charges of fraud and malpractice and all these kinds of things. But what had happened is, like we see back here, people in the US and the UK started to decline the MMR vaccination because they didn't want their children to become autistic. Okay, and so that has led to different types of measles outbreaks and things like that. Here just recently in the US, there was a few years ago a measles outbreak in that started in Disneyland and was actually passed around to 24 different states before it was all over. However, since 1998, there have been lots and lots of studies to try to find if there really is a link between MMR and autism, and there are none with credible research that have been found. In 2015, a study was published using 95,000 children, and that study showed no link between MMR and autism. Okay, so how are vaccines administered to a person? You can probably guess. One way is that they're injected. You get a shot, right? Others you can inhale. It used to be a few years ago, they would take the little syringe and you would inhale the vaccine up both nostrils instead of getting an injection for the flu shot. They've kind of backed off from doing that now, but that is one method. And then another method is through ingestion. The rotavirus comes in a liquid and it is taken by mouth. So what are vaccines? Okay, a vaccine is essentially an antigen that's administered to the body, whether injection, inhalation, or ingestion. And the body recognizes that antigen as foreign and starts to build an immune response to it and build immunological memory. Okay, so maybe jot that down. The purpose of vaccination, or what a vaccine does, is it administers or provides an antigen to the patient. Okay, it gets an antigen in the patient. The patient's immune system, its T and B cells, recognize and fight off that infection. The T and the B cells recognize and fight off that infection and build memory. So that would be your primary immune response, right? The first time hopefully, the first time you're exposed to vaccine preventable diseases is when you get exposed to that antigen through a vaccine, which is harmless or much less harmful than the actual pathogen. Then, if you ever do encounter that pathogen out in the natural environment, then your body has the memory built up and the immune system can attack and destroy that pathogen before disease signs and symptoms develop. So even if you have a vaccine and say you get, what would be a good one? Say you get the polio vaccine and you travel to Haiti, where they've had polio outbreaks before, and you drink the contaminated water, you consume the polio virus, and your body's like, oh, hey, I've seen that before. We're going to, destroy it. The virus still enters your body. You still become contaminated with polio, but the immune system wipes it out before it can set up an infection. Okay, so there's different types of vaccines, and that's one of the things that's on your review sheet for the exam. And the vaccine types could be what we call attenuated A vaccine, which has a weakened microbe in it, okay, it would have, oh, let me see. I think Hib is one of them. It would have that particular bacteria or virus, and it would still be alive, but it would be weakened. It could have a fragment of the microbe, like maybe a piece of the flagella that contains the antigen. You don't have to have the whole bacterium. You can just have harvest a bunch of flagella and inject those. For things like tetanus, botulism, diphtheria that cause issues because of toxins, okay, a vaccine might contain an inactivated toxin, a toxin that's not going to harm the cells, but elicit that immune response, or genetically altered pieces of microbes. And we'll talk about those here in just a little bit. One thing to always remember, though, with vaccines is that they do not provide immediate protection. Okay, so you go to the clinic, you get your flu shot, and tomorrow your roommate sneezes on your, computer or whatever, you touch it and, they had the flu, you'd still get the flu. It takes about 2 weeks for immunity to basically provide protection. Okay, so it's not going to be immediate. You have about 2 weeks before you have that protection there. And that vaccine, remember, stimulates that memory so that when you encounter the real microbe, the memory cells work very quickly. For most people, routine vaccination risks are pretty low. Now, like with any medical intervention, whether it's antibiotics or chemotherapy or physical therapy, any type of intervention has risks, okay? There's, no surgery, no type of intervention that we get from healthcare that is completely risk-free. Okay, now there may be some people that should not get vaccines, people that are immune compromised. There may be some vaccines that pregnant women shouldn't get. There are some that pregnant women do get, like the flu vaccine and also DTaP or Tdap. Some people with certain diseases and things like that may not be able to get vaccines, what may not be safe for them, but that's why we hear so much about what's called herd immunity, and that's where you vaccinate so many people in the population that they basically protect the unvaccinated people. They protect the people protects those who are too young or otherwise can't receive vaccines for other health reasons. Okay, so for example, when mom is pregnant, a lot of times they'll recommend that she gets the tetanus, diphtheria, and whooping cough vaccine while she's pregnant or very soon after birth, because baby is not going to be able to receive that vaccine until it's older. And so whooping cough is very, very deadly for young children. So if mom and dad and brother and sister all have the whooping cough vaccine, okay, the chances of them bringing it home to baby and being life-threatening to baby are very, very low. Okay, so that's an example of herd immunity, where the people that are around baby get the vaccine and help prevent the baby from getting it until it's old enough, to have its own vaccine. Healthcare or daycare workers should get the vaccine as well if they're going to be around young children. Okay, so here's just an illustration of that. So here's one infected person. I'm sure you've seen these with the coronavirus as well. Here's one infected person. you know, if nobody's immunized, the disease spreads rather quickly, okay? However, if some of the people are immunized, okay, you can see the rates of transmission are a little bit lower here, but if we have most people immunized, so say this is the baby or the person with the compromised immune system who is unable to get a vaccine, if everyone around them is vaccinated, that helps them become safer. Now, obviously, if this person and this person have direct contact, you know, just because everyone else is vaccinated doesn't mean that they'll be completely safe. But what it does mean is that this person isn't going to give it to this person, and then this person, and then this person comes in contact. Okay, second, third, fourth hand. Most pathogens require the vaccination of about 85% of the population to have effective herd immunity. However, measles and whooping cough is 95% because they're so very contagious. Let's see. And so public health initiatives really try to aim to get most people vaccinated to develop that herd immunity. Okay, so in the US, the CDC is what develops the recommendations of what to vaccinate for and when those vaccines should be given. And right now, CDC recommends vaccination for 15 different pathogens on a routine basis. And some of these pathogens need to have boosters, okay, or second or third or fourth shots after the initial one, or exposures to the vaccine after the initial one, to basically develop that immune system response that's adequate enough. And we'll see why sometimes we need boosters here in just a moment. But the CDC also has, if you look at this next slide, it kind of tells you, all right, for diphtheria, you should get dose 1 here, dose 2 here, dose 3 here, dose 4 here, and then 5th one here. The reason those are spread out, the reason that's different than maybe Hib down here, is because there's been a lot of research into that, and the developers, the people in the health industry know what time frames work best to allow the T and B memory cells to build up, and when they should be hit again to really develop the maximum immune response. So this is the guidelines are given here so that they're optimizing the time when these vaccines are being given to really help the person build up their immunity. Now, I'm not going to have you memorize when the 4th dose of DTaP is necessary or anything like that. I'm just going to use this as mostly kind of a FYI for your information. Some things that I know most of you are not parents, but some things that might be relevant to you as an adult, which I guess these are 11 to 12. But for those entering college, which you've already done, probably getting the meningococcal. And then, let's see, it doesn't have it on here. Getting your DTaP once every 10 years. Okay, that one only lasts for 10 years. That's pertussis, diphtheria, and tetanus. Okay, if you ever go and you have a deep puncture wound, you go to the doctor or whatever, they'll probably give you a tetanus shot if it's been over, just a couple of years, just to be safe, because tetanus is pretty nasty. Another one is influenza that you should get yearly. And I know some people don't like getting the flu shot. Nobody likes getting shots, to be honest. But I guess the way I look at it is, the more influenza virus antigens you can be exposed to through vaccination, whether it really helps a lot with this vaccine or this year's flu virus or not, I think is beneficial, because you never know what kind of crazy flu we're going to have 10 years from now. Remember antigenic shift and drift? Okay, the 1918 Spanish flu was a result of genetic drift, where we had some major recombination of antigens on the flu. And so if you've had exposure to all of these different types of antigens, the chances of you having immunity against perhaps a future flu might be beneficial, okay? So the more things you can expose your body to safely, I think, as far as antigens, I think the better protected you are. But everyone has their own feelings and opinions on vaccinations. Let's see. So we just kind of went over this. And then senior citizens, maybe if you have parents or grandparents, should get the shingles vaccine and also vaccine for bacterial pneumonia. And we talked about with the immune system, as you get older, your immune system isn't quite as potent as it was when you were younger. And so being protected from this pneumonia is going to be important as you get older. Okay, so different types of vaccines. Okay, so I kind of mentioned them a little bit earlier, but let's look at them a little more in depth. This might be a really good table for you guys to check out when you're studying. And then we'll have some other slides that go in more in depth here. But the first one is called, oops, that's a one, an attenuated vaccine, or attenuation basically means altered vaccine, okay? The virus or bacteria is alive. Okay, so the virus is active or the bacteria is alive, okay? But you're like, it's alive, what? But they have been rendered non-pathogenic. So they have undergone some sort of treatment that didn't kill them. It just removed or eliminated its potential to cause disease or to infect the body. Okay, so we'll talk more about that in a second, but some examples, MMR, rotavirus, and the chickenpox virus is those vaccines that are active or alive, if you will. And then over here on this side, these are the inactivated or the killed. Some of them are killed. Some of them are just portions of the virus or bacteria. Okay, these are not the actual bacteria themselves. So there's subunit vaccines, which are just a piece of the pathogen, like maybe one of those protein spikes on the outside of the virus, or it could be a piece of the flagella, it could be a piece of the capsule, just a piece that's been harvested. And then there's purified subunits. These have been basically genetically engineered. We've got hepatitis B, HPV, toxoid vaccines, which are inactivated toxins. So they take the tetanus toxin and damage it in a way so that it still has the antigen and promotes immunity, but it does not damage the cells. And then we'll talk about conjugate vaccines here in just a second. So live attenuated vaccines contain altered pathogens, okay? They don't cause disease, okay, but they're still infectious, meaning they can still reproduce. Or in the case of viruses, they can still replicate, okay? So why would the ability of the pathogen to reproduce or replicate be important in developing immunity? That's because you inject, say, 1 milliliter of vaccine. That's all that's introduced into the body. If that bacteria or virus is still active or alive, it can reproduce. And so remember, some bacteria can double their populations every 20, 30 minutes. And so if you're getting a harmless bacteria or virus replicating and producing more and more of itself throughout the body, just think how much stronger that immune response is going to be because it's detecting more and more of the antigen. Okay, so these are very beneficial. Let me pop up here, because it can really stimulate a potent immunological process and long-lived memory because of the pathogen replicating. Okay, so instead of just 1 ml of solution that's injected, now all of those cells perhaps could double every 20 minutes or so and just really stimulate that immune system. Let's see. Some drawbacks though. They could cause disease in a person that's immune system has been compromised. They could mutate back to the infectious form. And then the other thing is that they must be refrigerated because they're alive, and if they aren't held at the right temperature, they could negatively impact the benefit of the vaccine. Okay, again, this is things like MMR and chickenpox vaccines that are attenuated. Okay, so the rest of these are going to be inactivated, so they're not going to be able to reproduce. And one type of inactivated vaccine is the whole agent or whole pathogen is going to be delivered, but they're inactivated. Okay, so the whole attenuated virus is put in here, but it's still alive. Here, you put in the whole dead pathogen. Okay, some benefits. These are going to be safer and more stable, safer for immune compromised patients. But the drawback is that you'll need boosters to get the full immunity because they're not replicating. Okay, so you inject X number of cells or X number of virions, that's it. You're not going to get any more until you do the booster again later. So you inject the whole pathogen and they're inactivated by heat or chemicals or some sort of other process. Subunit vaccines are just, whoops, let me back up. These are going to be things like the polio vaccine given in the US and hepatitis A vaccines are whole unit vaccines. The whole virus is put into the vaccine. Subunit vaccines are just a piece of the antigen or a part of the pathogen. These are going to require what are called adjuvants. And the adjuvants, if you want to jot that down, is usually what people freak out about that's found in vaccines. You know, sometimes people are like, oh, there's mercury in the vaccine and everything. That's usually comes in the form of an adjuvant. And to be honest, the levels of mercury are so minute, it's not going to really be harmful. But some people don't want that injected into their body. But an adjuvant is going to enhance the immune response because the little pieces of the virus or bacteria that are injected are so minute, and there's a finite number of them. So those are adjuvants are given to help stabilize the vaccine and also strengthen the immune response. Let's see. Purified vaccines are basically harvested from a natural pathogen and then purified, or they're going to be basically a genetically engineered product. And we'll talk about recombination vaccines here in a minute, but things would be like HPV and Hep B. Toxoid vaccines are purified and inactivated toxins. Okay, so an antitoxin, we talked about that last chapter, like from an antiserum, like if you get bit by a rattlesnake, or something like that, you would be given the antitoxin, which is going to be antibodies that treat disease. Okay, so don't get those confused with the toxin, or I'm sorry, the toxoid. An antitoxin is antibodies that treat the disease of a sick person. A toxoid is going to be the vaccine, the preventative that you would take so you wouldn't get the disease if you happen to be exposed. Examples of toxoid is tetanus, toxoid, diphtheria are also put in, those two are put in together in a combination vaccine called DTaP. And then the AP, so the D stands for diphtheria, the T stands for tetanus, and then the AP stands for acellular pertussis, or whooping cough. Another subunit vaccine is called a conjugate or polysaccharide vaccine. And with some bacteria, the polysaccharide of the capsule, remember the outer coating outside of the cell wall of the bacteria, creates an immunological response. But the polysaccharide that's needed to identify that bacteria isn't powerful enough to get a good enough immune response. So a conjugated vaccine is going to be one where you have this polysaccharide or this sugar antigen, and you attach it to, or conjugate it to, or link it to a protein antigen. So it really just enhances the opportunity for that to be detected. Hib, meningococcal meningitis, and pneumococcal pneumonia vaccines are examples there. Some new vaccine techniques that are being developed and worked on is, there's two of them that we'll talk about, DNA vaccines and recombinant vaccines. A DNA vaccine is where they take the gene, the pathogen's gene, whether it's virus, bacteria, whatever, that codes for the antigen. Okay, so if it's a flagella protein, that's the antigen. Okay, the scientists will isolate the gene that codes for that protein, for that antigen. Okay, thinking back to our genetics unit, remember transcription, translation. Okay, the gene that codes for the transcription and translation of protein that produces whatever this antigen is would be harvested. It would be placed into a plasmid. Remember, in bacterial cells, we have our chromosome, and then we have the little other extra chromosomal DNA there, the plasmid. So they would take this gene, they would insert it into a plasmid, they would harvest these plasmids and inject the plasmid into the human host. Okay, the human host then could take up the plasmid, transcription, translation would occur, and then the cell, the host cell, the human cells become antigen producers. So the human cells are going to start producing whatever antigen it is, and the humoral, the B cells, and the cellular T cells are going to pick up on that and start to develop an immune response. And where they're really researching this is for HIV and certain types of cancer. Wouldn't that be cool if we had a vaccine against cancer? It'd be great. Well, we already have HPV, I guess. And then the other kind of newer area is the recombinant vaccines. And this is where we would still kind of do the same thing. Take the antigen producing gene from the virus. Okay, so here's the antigen, the little spikes here. Here's the gene that codes for the production of that spike. They would remove it, put it into a quote unquote harmless virus. And then they would inject this harmless virus with the foreign gene that produces the antigen into the person. And basically this harmless virus, or it could be a bacteria, goes around and replicates as it spreads from cell to cell and producing this antigen. Okay, so the pathogenic virus is not inserted into the body, only the gene that could make these little protein spikes. The body says, oh, hey, that's foreign, and starts to develop the immunity toward that. Okay, so this is where we're going to stop. The whole rest of the chapter we're going to skip. I'm not going to talk about the biotechnology, but I did want to just point out one thing. If we were in class, we would have done a blood typing lab. And I know most of you have probably done this in A&P class, or maybe done it in another setting. But I just wanted to talk about this really briefly because we did talk about blood types the other day. So blood type, okay, so this person, let's see. Do you know what type of blood this person has? Can you tell based on what this says? Type B, right? Has the type B antigens. Okay. So and I have type B blood. So if I were to do a finger stick and squeeze some blood out on the little blood typing card, and I come by with a solution, a little bottle of solution, and dumped on my red blood cells the antibody that attacks or attaches to type B, do you see how they attach there? What would form is a clump. Do you remember what a clump is called? Okay, this process is called agglutination. So if I were to put blood on a blood typing card and mix it with antibodies for type B blood, it would form a clump. If I were to put my blood on a card and put it with an antibody for type A, which has a different, which I wish they would have showed this a little bit differently, but it has a different shape there, they're not going to bind together, and there is no clumping. There is no agglutination. And so that's how we would have determined your blood type. You would have put your blood on, here's what the cards would look like. There's 3 circles on there. You'd put a couple drops of blood in each one. You'd put some A serum here, some B serum here, and then the RH factor serum there. And wherever it clumped, you have that blood type. Okay, so just as a review, think through here for a second. What if this clumped and this clumped, but this one didn't? What blood type would you be? You'd be A, B, negative. Do you understand? Clumping means you have that type. Okay, what if you clumped only the positive? It would be O positive. Didn't have a reaction, didn't have a reaction, but you did here. If none of them clumped, it'd be O negative. If this one clumped and this one clumped, you'd be A positive. Okay, so that's all I wanted to really say about the rest of this chapter. Okay, so don't forget mastering homework, and also test on Wednesday. Okay, so be in touch if you have any questions, just let me know. And test will be very similar to like what it was last time. You'll have a certain time length. When I get the test written, I will let you know what that is, but it should tell you in the directions. You have X number of minutes to complete the test, and it will need to be done on the 15th. Okay, you can do it at any time on the 15th as long as it's done and completed before 11:59 p.m. Okay, I think a couple of you still have not taken the SurveyMonkey survey found on last class period's daily checklist. So please do that. That will be very helpful for me. And let me know if you need anything or have any other questions. So have a good day, and we will talk to you soon.